Ebola is a single-stranded RNA virus discovered in 1976 in the Democratic Republic of Congo and is now primarily found in sub-Saharan Africa. Ebola is a zoonotic disease transmitted to humans from animals, likely bats or non-human primates, but the virus is now sustained in humans through person-to-person contact with infected blood, other bodily fluids, or contaminated objects. It is a highly pathogenic virus and can survive for up to 7 days on surfaces. The disease progresses from flu-like symptoms into gastrointestinal symptoms, eye redness, unexplained bruises and bleeding, and vomiting or coughing up blood. Organ failure, septicemia, and death can follow. Fatality rates vary from 25 to 90%. Ebola virus is a global concern as growth and urbanization can lead to increased transmission between individuals, and recent viral genomic sequencing suggests that Ebola does not hide in reservoir animals in between outbreaks until it infects a human, but it can exist as a persistent or latent infection in survivors of previous outbreaks who are unknowingly harboring Ebola, triggering new outbreaks when the virus reactivates.
So Ebola virus dysregulates the immune system. So in a normal defense, the innate immune cells would activate cells that help in getting rid of foreign pathogens. But in this case, the innate immune cells don't activate or don't help activate the T or B cells, which are part of the adaptive immune response. So in Ebola, the cells are infected and they participate in cytokine signaling that takes part in the immune system cell death and also causes for tissue death.
Dendritic cells are one of the first targets for the Ebola virus. Dendritic cells phagocytize the virus. Once the virus is inside the lysosome in the cell, it uses the NPC1 cholesterol transporter protein on the inner membrane of the lysosome to expel its genetic material into the cytoplasm. Like other viruses, cellular machinery is taken over and used for viral replication. Ebola inhibits the maturation of the infected dendritic cells, causing several problems. There is a downregulation of surface receptors like MHC2, CD80, CD86, and CD40, which not only results in a failure to signal that the cell is infected cells then fail to activate adaptive immune cells. Ebola virus also dysregulates cytokines. Infected dendritic cells produce decreased amounts of IFN gamma, which results in an inhibited immune response to the virus. The dysregulation of cytokines also cause infected dendritic cells to signal to nearby monocytes, macrophages, and NK cells, telling them to die.
The response that these cells take following infection is to produce inflammatory cytokines and chemokines. Macrophages and monocytes will increase their expression of pro-inflammatory cytokines like TNF-alpha, INF-gamma, and IL-6.
The pro-inflammatory response of the macrophages and monocytes signal for the endothelial cells of the blood vessels to become leaky, and production of IL-8 recruits neutrophils from the blood out through the leaky vessels to the site of infection. But neutrophils are not effective for clearing the virus. They end up causing more trouble than help. As neutrophils arrive, they degranulate, perpetuating inflammation, and in turn perpetuating more cytokine signaling to release more fluids from the blood vessels, allowing increased fluid and now blood to leak out of the vessels. Blood vessels become damaged as the pro-inflammatory response continues signaling to them, and it becomes a sort of positive feedback loop that the immune system contributes to rather than controls, calling for more inflammation, perpetuating more dysregulated cytokine signaling. The damaged vessels cannot deliver enough oxygen to organs, causing cells to die. Dying cells signal to nearby cells to increase inflammation, only exacerbating the problem, leading to organ failure. Ebola viral infection stimulates natural killer cells to behave strangely. Their population decreases as they are killed by the virus, but they can be found accumulating in areas of infection. And while natural killer cells are good for lysing infected cells, once outside of the peripheral organs of the immune system and now in tissues, They cause damage releasing pro-inflammatory cytokines like TNF-alpha and IFN-gamma, and they kill arriving lymphocytes, preventing infected cells from being cleared, allowing damage to continue. The mechanism for how the Ebola virus stimulates these behaviors in NK cells is currently unknown. Figure 1 is a depiction of some of the major signaling molecule profiles whose dysregulation is associated with cytokine storm and death in Ebola infections, IFN-gamma, IL-6, and IL-8 in particular. The immune system fights infection and maintains homeostasis with a regulated balance of pro-inflammatory and anti-inflammatory cytokines. But as discussed, Ebola drives cells to release an imbalance of cytokines, amplifying the dysregulation as it continues. The ability to maintain a stable range of signaling molecules at the onset of Ebola infection is associated with survival, while large changes in these cytokine and chemokine levels are associated with death. Looking at the graph, TNF-alpha shows a relatively stable presence in both non-fatal and fatal infections, but when looking at IFN-gamma, IL-6, and IL-8 over the course of infection, there's a significant difference in how much cytokine and chemokine levels change in non-fatal versus fatal infections. Non-fatal infections on the left show marked stability in IFN-gamma, IL-6, and IL-8 levels, indicating the immune system being able to maintain the crucial balance necessary to stop progression towards cytokine storm. In contrast, a wide range of change in cytokine and chemokine levels can be observed in the fatal infections on the right. Looking at changes in IFN-gamma, highlighted in red, the graph indicates stable levels in non-fatal infections. Compare this to the dramatically wider and unstable range of change indicated in fatal infections. The same is true comparing IL-6 highlighted in blue and IL-8 highlighted in orange. Again, non-fatal infections show a stable presence of both IL-6 and IL-8, while the fatal infections show a wide range of change or dysregulation.
This figure shows the percentage of lymphocytes between non-fatal and fatal cases of Ebola. Under these cells, we can see that an early humoral immune response is correlated to survival. We can see that the early response is critical to the activation of T cells. Looking under T cells, it indicates a steady production of T cells, NK cells, CD8 cells, T cells, and Ebola-specific T cells in non-fatal cases, while fatal cases show a continual decline as the infection progresses. Again, this figure indicates the loss of lymphocytes and the importance of an early humoral response to the likelihood of survival. We can see that day 5 is a critical point that separates survivors versus fatal cases, according to the percentage of IgM and IgG produced.
The normal job of the immune system is to eliminate infections, but when it's activated at a high level, it damages the host. So in Ebola virus, the dying cells are all releasing their contents into the blood, which triggers a cytokine storm. And in the Ebola, the cytokine storm sends an SOS signal that causes the immune system to disperse its cytokines, attacking the virus, but makes the blood vessel walls more permeable, so it leads to the leak of blood, which also causes the release of nitric acid, thinning out the blood and damaging the vessels further. All of these reduce blood pressure, which doesn't lead to death from blood loss, but instead from septic shock. We are saying that as the disease progresses, there is a loss of lymphocytes, and because Ebola virus first attacks macrophages and dendritic immune cells, while on the other hand, there's also an increase in macrophages, monocytes, and neutrophils that contribute to the inflammatory response. In the graph, you can also see that there's a decline in natural killer cells with the progression of the disease.